Hello, are there any questions? I apologize for being about a minute late. When I tried to open Zoom, it downloaded an update and I had to install the update, so I didn't know that was going to happen. All right, if there's no questions, I'm going to get started. Let me state that before we get started, I did a lot of grading this weekend, and I don't even remember all the labs I graded. But I think I've graded lab 00, lab 01, most of lab 2. Nope, only about half of lab 2. And most of lab 7. Now lab 7 has only been graded one time, and there's about a third of the students I need to get to. And then the other labs have been graded completely, meaning a second time. And I graded the last worksheet, which wasn't this week's worksheet. What the heck worksheet was it? Well, maybe it was this last worksheet. I did graded worksheet lecture 4B or lecture 4. I have not graded lecture 5, which I think was last week's. And I'll try and get to that soon. And then I graded 2 quizzes. Quiz, I'll talk about that later. Quiz 1A and quiz 2A have now been graded. And I went into quiz 1B, quiz 2B, and quiz 3B, and I awarded extra credit for each of those quizzes. One of the quizzes, I don't remember off the top of my head now, had an extra credit question. And so I gave everyone credit for that question. And I don't remember exactly what the problem was with quiz 2B, but I remember there was a problem. So I gave everybody, I think I gave everybody 3 points on quiz 2B. And then on quiz 3B, there was a question, I think it was the last question, that did not display the answers for at least 4 students. So I gave everyone 3 points of extra credit for quiz 3B. And I'm not sure why that question showed the question, but not the answers that you could select. And then it did that for, as far as I know, only 4 students. And then the rest of the students, it showed the answers. And I don't know what was going on there. All right, any questions about any of the grading?
Dr. Volpe,
Uh-huh.
can you put your headphones on?
Are you not hearing me?
It's hard to understand.
Okay, I've got it right here. I was going to wait until lecturing before putting it on, but I can put it on. Anyways, so I got a lot of grading done, and I hope to finish lab 7 tonight, but I've also got to finish the unknown project. If I get everything together, you'll receive results from 3 tests tonight on the unknown project. Any questions about any of that?
No.
Okay. Let me turn up the volume because I didn't hear that very well. Oh, that volume is pretty high. I'm not sure what the problem is there. All right. Let me shut down the grade book and display my screen. I think that one, I'll just back up. And then I can use that for the lab. All right, so why am I on here? I don't know what that is. Let's come back to here. And you should be seeing my screen now, which isn't showing you anything exciting, but all right, so we're supposed to be starting, actually we're already supposed to be in chapter 6 and then finish chapter 6 today. We need to finish chapter 5 and there's just no way we're going to finish chapter 6 today. So we're going to continue to be behind schedule. We have a lab for today, lab 8. And you should receive your unknown work or unknown project work today. And let me remind you, the infectious disease papers are due this Saturday at 11:59 p.m. Any questions about any of this? All right. Chapter 5. Remember, we have a number of slides to do yet. I think we had talked about this slide. The complete oxidation of glucose via aerobic respiration yields 36 to 38 ATP molecules in eukaryotes. Prokaryotes are a little more efficient and they generate 38 ATPs per molecule of glucose. And remember, you do need to know the summary equation for aerobic respiration, and that is glucose C6H12O6 plus 6 oxygen molecules and 38 ADP and 38 inorganic phosphates yield 6 carbon dioxide molecules, 6 water molecules, and 38 ATP molecules.
Dr. Volpe.
Uh-huh.
Sorry, there's some background noise.
I'm sorry, what?
I'm hearing some background noise. I don't know if anybody else is.
Is anyone else hearing background noise?
Yes,
hearing
there's a
it
lot
too.
of background feedback noise.
I'm not sure why you're getting feedback noise. I've got my, let me turn down the volume, see if that helps. I've got the headphone on.
That
I
seems
don't
to be
hear
better.
background noise here. That's the wrong one.
It seems better now.
It's better now?
Yeah, thank you.
It might have been that the headphone volume was too loud. When you're talking to me, you're going to have to talk loud with the headphones on. I don't hear you guys too well. I'm not sure why that is the case. So if you want to say something, you need to get close to the microphone on your computer. Because I turned down the volume and that does, you said, sounded better, and I don't know why that's the case. All right, let's move on. So this is showing you aerobic respiration in prokaryotes. You do need to know the numbers, need to know the numbers of ATP made in each step, glycolysis 2 ATP, the Krebs cycle 2 ATP. We're talking about per molecule of glucose. And then 34 molecules in the electron transport chain. You do need to know the other steps along the way too. All right, so we talked about aerobic respiration. It is the most efficient way to make ATP. But you have to have oxygen around to use aerobic respiration. There are other organisms that use anaerobic respiration, where the final electron acceptor is not oxygen. It is another molecule other than oxygen. And there are many different final electron acceptors in aerobic respiration, depending on which form of anaerobic respiration the bacteria is performing. It can be, well, it's almost always an inorganic molecule, such as nitrate and sulfate, but it could be iron. In anaerobic respiration, usually the anaerobe does perform glycolysis and the Krebs cycle, and they do have an electron transport chain. However, that electron transport chain is shorter in anaerobic respiration, so the yield of ATP is less than for aerobic respiration. And the yield of ATP in anaerobic respiration is someplace between 2 ATP per molecule of glucose and 36 ATP per molecule of glucose. So it depends on which form of anaerobic respiration. Some of them are very efficient and are almost as efficient as aerobic respiration, like the ones generating 36 molecules of ATP. And I'm not sure how low the lowest is, but it's not much better than fermentation, which we'll talk about next. Because anaerobes generate less ATP molecules per molecule of glucose, they tend to grow more slowly than aerobes. Any question about any of that? All right, so there is actually aerobic respiration. Let me state that it is generally the same, but just this electron transport chain is shorter. And then we're comparing that to fermentation, which is another process using catabolism to generate energy, and then use that energy to make ATP. Glycolysis precedes the fermentation reaction, and it's the same glycolysis we discussed, where glucose is made into 2 pyruvic acid molecules. There's a net gain of 2 ATP molecules, and then we generate NADH and H+ from the oxidation of glucose. Now you can oxidize other molecules besides glucose, but we'll talk about it mainly with glucose, the same as we did with aerobic respiration. So we have glycolysis generating 2 ATP and 2 NADH, the same as before, and the glucose is converted into 2 pyruvic acids. The pyruvic acid is then metabolized in 1 or 2 steps by a fermentation reaction or 2 reactions, making the fermentation end products. And then that NADH that is made in glycolysis is used up by the fermentation reaction. The final electron acceptor in fermentation is an organic molecule where the electrons flow to, like pyruvic acid, which is then converted into the end products of fermentation. Now here's a question for you. Why does a cell that's making ATP by fermentation, why does the cell generate NADH in glycolysis and then use that NADH in the fermentation step rather than just keep the NADH around until aerobic respiration can happen. Because NADH, if you remember, carries the electrons to the electron transport chain, and then those electrons are used to make ATP. Why does like your muscle cell, when it's performing fermentation, not just keep that NADH around and wait until oxygen comes back to the muscle cell and then use that NADH with electron transport chain? Because if the muscle cell did that, it would get a lot more ATP than it would get from fermentation.
Is it because it needs immediate energy? When
It's
oxygen is
because
not
it-
available?
Go on.
When oxygen is not available, it will convert it to lactic acid, and then
Yes. So when oxygen is not available, your muscle cells perform fermentation because they can't perform aerobic respiration because oxygen is ran out in the muscle. And to keep working, the muscle uses fermentation and makes ATP and glycolysis. And then it uses this NADH made in glycolysis to make lactic acid. And then that regenerates the NAD plus, which can continue to be used in glycolysis. So if you run out of NAD plus, glycolysis cannot happen. And then you have no ATP being made in the muscles. And if you ever watch the Olympics, usually during one Olympic event, there's at least one athlete that they're running along and they just collapse. What happens is their heart and their lungs are beating like crazy to try and give the, I guess I should say working like crazy because the lungs don't beat. They're working like crazy to bring oxygen to the muscle, but they can't do it fast enough. And so the muscle runs out of oxygen. Rather than stop working, which it would have to if it had no ATP, the muscle falls back on fermentation and it makes a little bit of ATP. But once the muscle runs out of glucose, the muscle cannot perform fermentation and no ATP is made. And that Olympic athlete just collapses because their muscles cannot work. In the wintertime, the coach runs out and throws a blanket over them and they're perfectly fine. They'll get up after the heart and the lungs have brought oxygen to the muscle, and then their muscle can perform aerobic respiration. And the heart and the lungs, well, actually the heart brings glucose to the muscle again too. And they'll get up and then walk across the finish line. I'm sure you've seen that. Like I said, it usually happens at least once in the Olympic athletes. I think in the summertime, the coach runs out and this sprays down the athlete with water. Okay, any question about any of that? All right. So fermentation is where glucose is converted to pyruvic acid by glycolysis. And then in one or two fermentation reactions, the pyruvic acid is converted into the fermentation end product. We're going to talk about two fermentation reactions, lactic acid fermentation, which your muscle cells can do. And then most cheeses use lactic acid fermentation. Many fermentation reactions perform lactic acid fermentation, like making yogurt and sauerkraut. And then another fermentation reaction we're going to talk about is alcoholic fermentation, which we have two fermentation reactions. you don't need to know we first make 2-acetylaldehyde, and then that's converted to ethanol in alcoholic fermentation. In both fermentations, lactic acid fermentation, which that also happens in sourdough bread. But it's more commonly discussed in sauerkraut or making cheddar cheese or most other types of cheese. And anyways, lactic acid fermentation happens in one step, making lactic acid. And then alcoholic fermentation happens after glycolysis. It happens in 2 steps. CO2 is given off right there. And that gives beer its fizz. Wine is made in an open barrel casket or an open bucket or something like that. And the CO2 usually vents to the air. But if you were to cork the wine bottle, we call that champagne. Okay. And then in the second reaction, ethanol is made. All right, any questions about lactic acid fermentation and alcoholic fermentation? There are different end products and the different fermentation reactions, and there are 1, 2, 3, 4, 5, 6 fermentation reactions. We've only talked about 2. Like I said, lactic acid fermentation is the most common. Yeast does alcoholic fermentation, and that is made in most bread other than sourdough bread, which sourdough bread usually has both going on, lactic acid fermentation and ethanolic fermentation. And then beer and wine, making that uses alcoholic fermentation. Here's one. Propionic acid fermentation. This happens in Swiss cheese. And then there are other fermentation reactions that make other products, and they can be used in industry to get certain products. Like this is, you don't need to know this table, but it tells you what the end product is. Ethanol, which I forgot to mention, that's used to make fuel, which we take that ethanol and put it in our car for adding gasoline, ethanol to gasoline. And it usually uses a fruit juice to like make wine or a malt extract to make beer. It's lactic acid fermentation makes a lot of fermented products like sausage, sauerkraut, rye bread, most cheeses other than Swiss cheese. And usually you use milk for that, but it can be other things like for rye bread, for example, it's rye flour. There's acetic acid fermentation that makes vinegar. You usually use wine to make that ethanol, to make the vinegar. And then there are other products that are made in industry, like acetone and butanol, glycerol, methane. So a summary of aerobic respiration and anaerobic respiration and fermentation is shown here. You should know the reaction. You should know the growth conditions. Aerobic respiration has to happen in oxygen, meaning it's an aerobe, but anaerobic respiration and fermentation happen with anaerobes, and fermentation can happen in both aerobes and anaerobes. Okay. You should know the final electron acceptor. It's oxygen for aerobic respiration. It's another molecule other than oxygen for anaerobic respiration. And you don't need to know for fermentation, but it's an organic molecule, usually of glucose metabolism. You don't need to know that column. You should know how much ATP you make. For aerobic respiration, prokaryotes make 38 and eukaryotes make 36 to 38. I think we talked about that earlier, didn't we? Nobody's commenting. Anaerobic respiration makes fewer than 38, but more than 2. And then fermentation makes 2 ATP. Did somebody start to say something?
Yes, we talked about that.
Okay, great. All right. So on this slide, I'm going to tell you, we talked about carbohydrate catabolism and how glucose is metabolized to make ATP. Well, you can use other carbohydrates to make ATP. Usually the other carbohydrates are just broken down and either broken into glucose or converted into glucose and then sent down aerobic respiration in the normal way, starting from glucose. But you can metabolize or catabolize, I guess I should say, other food products, including lipids and proteins. There is a problem for this because large molecules cannot cross the cell membrane. So a lipid molecule, well, if it's really large, it's going to have a hard time getting into the cell just because it's so large. A protein molecule cannot be brought into the cell, and it will not get through the cell unless there is a transport protein molecule. Usually what happens with lipids and proteins, the cell will digest the lipid and the protein into the monomers outside the cell. And that's shown here. The microbes will produce lipases and proteases, digest these molecules extracellular using enzymes secreted outside the cell, and then bring across the monomers, which for lipids will be glycerol and fatty acids. For proteases, it'll be amino acids. And that's the same way that we digest lipids and proteins. We digest them extracellular, outside the cell, and then bring the monomers inside the cell. Let me warn you or notify you that amino acids require an extra step to digest them. The amino group has to be removed. And usually when we do that, we'll just convert that amino group into urea and then we'll pee it out. But amino acids are a little more complicated, just one more step complicated to digest. And then the amino acids and the monomers of the lipids are converted so that they can enter the Krebs cycle. All right, any question about any of that? So this slide is showing you that carbohydrates usually are converted into another sugar or into glucose, and then that's converted into glucose, and then it's sent down the aerobic respiration in the normal way. Proteins, depending on which amino acid it is, can be converted into pyruvic acid and then sent down aerobic respiration the rest of the way. But some amino acids are converted into acetyl coenzyme A and then enter the Krebs cycle. Other amino acids are converted into an intermediate of the Krebs cycle and then enter the Krebs cycle. Lipids are always broken down into glycerol and fatty acids. The glycerol is converted and joining the glycolysis midway through glycolysis. The fatty acid is broken down into 2 carbon chunks and then converted into acetyl coenzyme A to enter the Krebs cycle. Any question about any of that? So aerobic respiration is the most efficient way to generate ATP. And then ATP will be used by the cell for various cellular processes, such as active transport, moving a molecule across the cell wall or cell membrane, against its gradient. That costs ATP. For movement of the cell, both for a cell that has flagella, as well as for our muscle movement. And then we'll use ATP for synthesis of new cellular components to make an anabolic molecule. We need to spend ATP to make the molecule larger in anabolism. Most cellular ATP, at least in our cells, is used for the latter, to make larger molecules, such as the biosynthesis of protein, or the biosynthesis of lipids, polysaccharides, and nucleic acids, for all of the 4 molecules of life. Now, I've talked about anabolism and catabolism as separate and distinct pathways. However, they are integrated. There are common intermediates, and we call the pathways amphibolic if in the metabolic pathway, the pathway can move in both directions. The amphibolic pathways bridge the pathways involved in carbohydrate, lipid, protein, and nucleic acid metabolism. So this chart is showing you some of the amphibolic pathways that our cells have. And as you can see, the Krebs cycle, all of this is amphibolic, meaning it can go this way, but we can go the reverse way. And it's not showing all of the amphibolic pathways because it's showing the breakdown of lipids. But as you know, if you were to eat too much carbohydrates, you're not going to store it down as carbohydrates. It'll be converted into a lipid and then stored as fat. So this can go this way, where you eat too much carbohydrates, it'll be broken down into acetyl coenzyme A and some product of glycolysis, and then converted into glycerol and a fatty acid. The fatty acids will be joined with the glycerol to make the lipids. Any question about any of that? So the important thing to realize is that catabolism and anabolism are interconnected. Even our cells, anabolism and catabolism are interconnected. And our cells are lousy at interconnecting anabolism and catabolism. Green plants are much better at connecting the two, anabolism and catabolism. Any question about any of that? And I should say microbes are much better too. I said green plants because you know them better, but actually microbes are very good at integrating metabolism. Our cells are not so good. All right, any questions about the integration of metabolism? If not, let's move on to our last topic, photosynthesis, the conversion of light energy into chemical energy. The chemical energy may be used to convert atmospheric CO2 to glucose. And we do need CO2 and water to make glucose. This reaction is called carbon fixation, where you fix carbon dioxide and water into glucose. Photosynthesis is performed in obviously green plants, you all know that, but it's also performed in algae, and then the cyanobacteria, the photosynthetic bacteria. And strictly speaking, this is a family. There is more than one family that performs photosynthesis in the bacteria. This mechanism for fixing light energy into chemical energy and potentially making glucose is synthesizing organic compounds. So this is anabolism. And without photosynthesis, animals and microbes would not have food to consume or to eat for humans and animals. Only photosynthetic organisms can engage in photosynthesis. These organisms contain a light trapping pigment. One such pigment you know is chlorophyll. Light releases electrons from chlorophyll, and then the light energy is converted into chemical energy via an electron transport chain. And we'll talk briefly about that. The light energy is converted into the movement of electrons, which then move down an electron transport chain. And the purpose of that is to make ATP. NADPH is also generated along the way. Now ATP and NADPH may be used up to synthesize glucose from CO2 and water. Any question about any of that? There are two types of photosynthesis. Now this is probably new to most of you. There is cyclic photosynthesis and non-cyclic photosynthesis. Non-cyclic photosynthesis is the one you are probably familiar with. A summary equation of non-cyclic photosynthesis is you get 6 carbon dioxide molecules, 12 water molecules in the presence of light to make glucose, C6H12O6, 6 oxygen molecules, and then 6 water molecules. Sometimes you'll see it written out where they subtract this and then reduce 6 water molecules here. But I prefer this reaction shown here, because if we look at where all of the 12 oxygen atoms come from, 6 times 2 is 12. All 12 of these oxygens come from water. And so I like putting 12 water over here, because that explains where all of the oxygen atoms in oxygen from photosynthesis come from. Note that none of the oxygen comes from carbon dioxide. Okay, and that's the same as aerobic respiration, where we start with carbon dioxide and go that way. And all of the water comes from glucose. Is that right? Sorry, all of the carbon dioxide come from glucose. None of the carbon dioxide comes from the oxygen. All of this oxygen is converted into water. There is another photosynthesis reaction, and let me just say that there is an anaerobic photosynthesis reaction that occurs by some phototrophs. And oxygen is not generated. That's why it's called anaerobic photosynthesis. You don't need to know this. We're not going to talk about it further, so don't memorize it. You should know, however, that besides non-cyclic photosynthesis, we also have cyclic photosynthesis. In cyclic photosynthesis, the cell only makes ATP from ADP and inorganic phosphate. Any questions about any of that? All right, if there's no questions, let's move on. Generally speaking, all cells can engage in both cyclic photosynthesis and non-cyclic photosynthesis. Like in your house plant, it has a cell. Whenever that cell wants ATP, it will engage in cyclic photosynthesis. However, if that cell wants to make glucose, it'll engage in non-cyclic photosynthesis. Remember, in non-cyclic photosynthesis, you make ATP, but that ATP is used up to make the glucose. All right, in cyclic photosynthesis, we have light, which strikes the chlorophyll, and that makes the electron move out of the chlorophyll. The electron moves to this protein in the membrane, to that protein in the membrane, to that protein in the membrane, and then back to this protein, which contains the chlorophyll. And the electron actually does move back to the chlorophyll molecule from where it started. And that's why it's called cyclic photophosphorylation or cyclic photosynthesis. The electron moves in a cycle, ending up where it started. As this electron moves through this protein here, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. That hydrogen ion gradient is then used to make ATP, that the excess hydrogen and positive charge on this side will want to move to that side, and they can only move through an ATP synthase protein, and they give this protein the energy it needs to make ATP. This should sound familiar to you because this is similar to the way that our cells make ATP from aerobic respiration. The electron moves in aerobic respiration from glucose, and then it moves through either Krebs cycle or glycolysis to NADH, which then carries it to the proteins in a membrane that we call the electron transport chain. And some of those proteins that get the energy they need when the electron moves through it to pump hydrogen ions from one side of the membrane to another. Now in eukaryotes, aerobic respiration happens in the mitochondria. In eukaryotes, photosynthesis happens in the chloroplast. Where do you think photosynthesis happens in prokaryotic cells? It can't happen in the chloroplast because there is no chloroplast in a prokaryote. What?
In the cell membrane?
Yeah. Photosynthesis occurs in the cell membrane. So this is going to be the membrane of a chloroplast of a green plant cell, or in a bacteria, this is the cell membrane. All right, any questions about any of that? And this slide is showing you that the energy source, the donor of the electron, can vary. In aerobic respiration, it's glucose is the energy source, the donor, the electron donor. For photosynthesis, the electron donor happens from sunlight striking the chlorophyll, and then the electron moves out of the chlorophyll. The final electron acceptors can also vary. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is a molecule other than oxygen. And then in an organic chemical compound or in fermentation, the final electron acceptor is a organic molecule. For photosynthesis, the final electron acceptor is actually the chlorophyll. All right, any question about chapter 5? Now, if not, let's begin chapter 6. Chapter 6 is discussing microbial growth. Of course, know the terms, understand the various growth requirements, understand that growth goes through 4 phases of growth, and be able to compare the different methods for measuring microbial growth. When we're talking about microbes and we talk about growth, we're referring to an increase in the number of cells, not an increase in the cell size. And so that differs then with children when we're talking about growth. We're referring to mostly the increase in their size, not their cell size, but their body size. And I've got a little video here showing you the growth of cells. And this will take a little while to load, so I'll continue while it's loading. There are different requirements for microbial growth. There are both physical requirements and chemical requirements. For physical requirements, that includes things like there has to be the correct temperature, the pH, the correct osmotic pressure, or the salinity for the growth to occur. Outside of the correct temperature, you cannot get growth. And the correct chemical sources, all living things need carbon, nitrogen, sulfur, phosphorus, trace elements. They all have oxygen requirements, even if it's absent. That is an oxygen requirement. Some need oxygen, some need a lack of oxygen. And then there might be organic growth factors that are a chemical requirement for growth. Any question about any of that? If not, let's go to our little video.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any question about that little video? If not, we'll move on. So each species of microbe has its own minimum growth temperature, maximum growth temperature, and optimal growth temperature. The optimal growth temperature is where the species will grow its fastest, and the minimum temperature is the temperature at which the species grows slowly. But if you lower the temperature any further, the species will not grow. Any question about any of that? We classify microorganisms on the basis of their preferred temperature range. There are psychrophiles. These are organisms that grow best at cool temperatures like your refrigerator, and they grow best near 10 degrees Celsius. There are psychotrophs, and they grow best between 20 and 25 degrees Celsius. These would be things like microbes you find in the soil. There are mesophiles, and they grow best around 37 degrees Celsius. All human pathogens are mesophiles. There are also thermophiles, and they grow best between 60 and 65 degrees Celsius, which is really hot. And then there's hypothermophiles, and they grow best between 90 and 100 degrees Celsius. Remember, 100 degrees Celsius is the temperature that water boils at. Now, you should know that each organism has its own classification, that the mesophiles is not rigidly defined. Not all mesophiles grow best at 37 degrees. For example, Staphylococcus epidermidis grows best at, I think it's 30 or 31 degrees Celsius. And we do consider it a mesophile. Why does Staphylococcus epidermidis not grow the best at 37 degrees if it is a mesophile? Come on, you guys know this. Where does Staphylococcus epidermidis grow?
On the skin?
On your skin. And your skin is a little cooler than 37 degrees Celsius. And so Staphylococcus epidermidis actually has its optimal temperature a little lower than 37 degrees. Okay, you got that? But we still call it a mesophile. And the range also varies for the minimum temperature a species will grow, and then the maximum temperature a species will grow. The point is that this graph is not rigidly defined. Any question about that? For example, this shows that psychophiles can grow down to -10 degrees Celsius. Well, if that psychophile did not make antifreeze within the cell, it could not grow below 0 degrees because the cell would freeze. And then the cells which are growing below 0 degrees, these are cells that are putting antifreeze or some chemical that acts as an antifreeze into their cells. Any question about any of that? All right, I want to talk just briefly about the food spoilage. Food spoilage occurs best when food is left at, well, about 60 degrees to 130 degrees Celsius. And that's about 15 degrees to maybe 52 degrees Fahrenheit. This is when the organism can grow its most rapid, and it'll spoil food more quickly than food placed at a lower temperature. Now note that most bacteria can grow at, well, grow slowly in the refrigerator and then above the optimum or the most rapid growth region. Meaning you can get an organism growing above 50 degrees, but it's going to grow slower. And you can get an organism growing less than, well, 10 degrees or less, but it's going to grow slower than if it was growing, or whatever I said, 15 degrees Celsius to about 52 degrees Fahrenheit. Food spoilage occurs usually either when it's inappropriately handled and then allowed to sit out for too long, or when it is packaged in too large a volume and then put into the refrigerator. The problem with this is that the food doesn't get cool enough fast enough. And in this region here, which is several hours long, actually it goes down even further, down to about here, the organism can grow. So we don't want to put food in large containers in the refrigerator. You want to have it, the food cool quickly enough so that the organism cannot grow. Any question about any of that? All right. Let's move on to talking about physical requirements, the pH. Most bacteria grow best between pH 6.5 and 7.5. Most of your lab cultures will grow best at that temperature, and away from that temperature, they will grow slower, if at all. Most yields, muslin yeast grow a little bit more acidic than most bacteria, and so they grow best around pH 5 to 6. But there are acidophiles, microbes that grow in very acidic environments. And that would be a pH between pH 0 to pH 3. And volcanic environments might be a place where we find acidophiles. Where in your body do you have acidophiles growing?
In the stomach?
Yes, in your stomach. This pH of the stomach is between pH 1 and about pH 3, assuming you haven't eaten anything. Okay. If you eat something, the pH of the food will lower the pH of the stomach. Excuse me, not lower the pH of the stomach, it'll raise the pH of the stomach. All right, any questions about pH? If not, let's move on to osmotic pressure. Most microbes grow best or optimally at a certain salt level or a certain molasses or sugar level. And beyond that range, the microbes will grow less well. And way beyond that range, they won't grow at all. High osmotic pressure can remove the necessary water from a cell. Like here, I'll try and blow that up. Here we have a cell in an isotonic solution, and the water inside the cell is equal to the outside. And if we put this cell in a hypertonic solution, water will then leave the cell, resulting in plasmolysis, where the cytoplasm will pull away from the cell wall and then will shrink. It's generally a little bit longer than the, little wider than the nucleus, but that's usually what stops it from shrinking. And when this happens, the cell is likely to die because it doesn't have enough water. Humans have actually been using high osmotic pressure to preserve foods for millennia, such as when we salt meat or we have molasses. It's just pure molasses, it doesn't spoil. And as I mentioned, the bacteria that fall into the molasses bottle will go through plasmolysis as water leaves the cell. Another example would be salted fish is using high osmotic pressure to keep the cells from growing, and then that keeps the food from spoiling. And if you don't know, cabbage, for example, will last a lot longer with sauerkraut, which is made of cabbage, than it would if you were to just use the cabbage leaves or whatever, a cabbage directly. That's what I'm trying to say. Did I confuse anyone on that? I'm not hearing anything, so hopefully you understood what I was trying to say. All right, organisms that love to grow in salt, we call halophile. Halo, salt, phile, lover. So literally it means lover of salt. But what we mean is it likes to grow in salt. There are obligate halophiles, and they require a high salt concentration in order to grow. And if they get out of this high salt concentration, they will not grow. Any question about any of that? There are also facultative holophiles, and they do not require high salt concentrations to grow, but they can tolerate high salt concentrations to grow. And so we can get them to grow easily up to 15% sodium chloride. There are holophiles that grow much higher than that. I guess the halophiles on your skin can grow up to 20% sodium chloride, but all of them are facultative halophiles. They do not require that high of salt to grow, and they can grow at 2% sodium chloride or less. An obligate halophile requires a high salt concentration to grow, and they actually die or fall apart if they're not in that high salt concentration. And that could be some organisms living in the Dead Sea, which I think it gets up to 30% sodium chloride in the Dead Sea. If you don't know where the Dead Sea is, it's between Palestine and Jordan. In a hypotonic solution, this is low osmotic pressure, water will enter the cell by osmosis. And if the cell doesn't have a cell wall, the cell will lyse. Its water continues to enter the cell, and then the cell membrane will eventually pop. Now, as I mentioned, the cell wall will prevent water from entering the cell and popping the cell membrane, but only to a point. Once the cell membrane expands to touch the cell wall, no more water can enter the cell. Any question about any of that? All right. Another chemical requirement for living organism is all living things require carbon. Typically, the dry weight of cells or organisms is about half of the dry weight. So regardless of what microbe you're talking about, or whatever organism you're talking about, half of the dry weight is from carbon. Carbon is used in structural organic molecules, such as carbohydrates, proteins, but it's also an energy source. As you know, we digest glucose that has 6 carbons in it. And then carbon is, for heterotrophs, use an organic carbon source like glucose. And then the autotrophs can use an inorganic carbon source, such as CO2, dissolved in the air or the water. Any questions about any of that? Sorry, I'm zipping up my coat here and making a lot of noise doing that. Some other chemical requirements for living organisms, they all require nitrogen, sulfur, and phosphorus. Nitrogen is 14% of the dry weight for most organisms. Nitrogen is used in amino acids, protein, DNA, RNA, and ATP. Nitrogen is largely obtained by cells from the decomposition of proteins, but you can get it from other sources, like many bacteria can use ammonium, so they can pick that up from the soil, or they can use nitrate, NO3 with a negative charge, or some other nitrogen source. A few bacteria can even use nitrogen gas in the atmosphere, and these we call nitrogen fixing bacteria, because they can pull nitrogen out of the air and then make it available to other organisms. And initially that organism pulls nitrogen out of the air, and then we'll use that nitrogen to make amino acids, proteins, DNA, RNA, or ATP. But when this organism dies, these biological molecules will be decomposed, and that will make nitrogen available to other organisms. Any question about any of that? All right, sulfur is largely obtained from amino acids, but you can get it from thiamine and biotin 2B vitamins. Sulfur is needed in DNA and RNA synthesis. Sulfur is largely obtained from the decomposition of proteins, and that is how we get most of our sulfur needs. We can get it from mushrooms as well, but mushrooms don't have hardly any protein. Many bacteria can get their sulfur needs from sulfate, which green plants can do. This would be toxic to us, but it's not very toxic. It's just slightly toxic. And then some bacteria can get their sulfur from hydrogen sulfide gas. And this to us is very toxic. Organisms also have a phosphorus need. Phosphorus is needed in DNA, RNA, and ATP. Phosphorus is also in the membranes of cell, such as the cell membrane. And we talked about this before. A phospholipid is the main molecule of a membrane. And obviously it contains phosphorus in it. Any question about any of that? All right. Living life forms also require trace elements. Trace elements are inorganic elements required by microbes in very small amounts. Trace elements are often required by enzyme cofactors, meaning the enzyme will not work unless the trace element is around. Some examples of trace elements include iron, copper, molybdenum, and zinc. For our cells, we need to have iron, copper, and zinc in trace elements, or else we would eventually get sick and then eventually die. Usually, the trace elements are needed in such small amounts that the organism can get them, the trace elements, from their environment. And that is how we get copper, zinc, and maybe molybdenum. Okay. And then most people can get iron in the food they eat and the liquids they drink, especially most men. It is true that menopausal women during their menstruation cycle or right after, that they may not be getting enough iron in their diet, in which case you need to supplement the diet to be fortified with iron. Any question about any of that? The last chemical requirement I think I want to talk about, well, I guess there's oxygen, so it's not the last. That is required by many cells to grow are organic growth factors. Organic growth factors are biological molecules, meaning they're an organic molecule, but they're obtained from the environment of the organism. So the organism cannot synthesize an organic growth factor, but they're a biological molecule. They're not an element. Remember, an element is just an atom, like sulfur well, like iron, for example, we get that as actually as atoms. Same with copper and zinc. Sulfur is usually bound to oxygen on the planet Earth, unless it's in a biological molecule. And as I stated, for us, we get most of our sulfur needs from the meat we eat. So some examples of organic growth factors include vitamins, like vitamin C. That's an organic growth factor. Certain amino acids are organic growth factors, like the amino acid lysine is an organic growth factor for us. And then some organisms, usually microbes, require purines or pyrimidines, meaning the molecules of aerobic respiration. For organic growth factors, you don't usually find many of them in the food you eat, although meat would be an exception. Meat would offer purines and pyrimidines and certain vitamins. Any question about any of this? All right. Molecular oxygen is another requirement for growth, and some organisms require it, aerobes, and then other organisms do not require it, and most of them prefer not to have it around. So molecular oxygen is, in a sense, a toxic substance to us, as well as to all living cells. And there are 4 toxic forms of oxygen. Usually it's not O2 itself, it's some metabolic component of oxygen, meaning the oxygen is metabolized, and then there will be an intermediate, which can be toxic to us. And usually the toxic molecules are made in low concentration, like the enzyme didn't quite work correctly. So instead of making the correct molecule, we made ozone. And that is one way that Portland and Vancouver get greenhouse gas emissions and pollution. That's the word I'm trying to look for. Get pollution. And that's because they are metabolizing the oxygen. All right, any questions there? Let's briefly go through the 4 toxic forms of oxygen. Like I said, oxygen gas is not toxic, but singlet oxygen is oxygen gas, meaning the oxygen molecule, where the electrons are boosted into a high energy state. That makes this singlet oxygen, the oxygen molecule, extremely reactive. An example of singlet oxygen is the lysosomes of your phagocytic white blood cells. They have singlet oxygen in there because this is so reactive that it tends to kill, we'll say bacteria, all microorganisms, but it tends to kill bacteria. Any questions about singlet oxygen? Okay, I think that talking is from another room, so I don't think that's anyone here. Superoxide free radicals is also a toxic form of oxygen. Whenever you have a free radical, it's generally bad for cells. A free radical is a molecule that has an extra electron, but this electron isn't paired. That gives oxygen here a negative charge, because oxygen gas is neutral, and if you add a extra electron to it, the oxygen gas will now have a negative charge, and this will be a free radical. Free radicals are bad for cells because generally speaking, whatever a free radical will bump into, it will steal an electron and then damage what it bumped into. Oxygen radicals are reactive because they have a single electron, and a single electron is not stable. This molecule will want to gain another electron. Or conversely, it could lose this electron. Okay? But 2 electrons are stable, 1 electron is unstable. And molecules with 1 extra electron are called free radicals. Free radicals are highly unstable. They are made in aerobic respiration when oxygen is used as the final electron acceptor. So all of our cells have to tolerate superoxide free radicals. And we do have some help that ATP is happening in the mitochondria. And as long as the superoxide free radical stays in the mitochondria, that's not going to damage the rest of your cell. We also have enzymes for converting the superoxide free radicals into something that can be made less toxic or untoxic. For example, 2 superoxide free radicals are combined with 2 hydrogen ions in the presence of the enzyme SOD, which is superoxide dismutase. And that will generate hydrogen peroxide and oxygen from the 2 superoxide free radicals. Any question about any of that? Our cells live in oxygen, so we have SOD, that enzyme. Well, I was expecting somebody to ask, well, isn't hydrogen peroxide toxic to cells? Yes, it is. And that's actually my next slide of a toxic form of oxygen, peroxide anions, which include hydrogen peroxide. So peroxide anions can be combined with hydrogen and oxygen to form hydrogen peroxide. And then the hydrogen peroxide is neutralized by the enzyme catalase. We'll talk about this a little bit in the lab. Hydrogen peroxide, which is 2H2O2, not H2O, but H2O2, in the presence of catalase, is made into 2 water molecules and 1 molecule of oxygen gas. And how you can test for catalase is you just add it, and then this oxygen that's generated will create bubbling in the hydrogen peroxide. Some cells live in oxygen but don't have the enzyme catalase. And what they are relying on is the enzyme peroxidase, which can convert hydrogen peroxide and 2 hydrogen ions into 2 water molecules. All organisms that live in oxygen have to have either catalase or peroxidase, and a few cells even have both enzymes. Any question about any of that? All right, the 4th toxic form of oxygen is the hydroxyl radical. It is OH, that group, or the hydroxide anion, but it has an extra or an unpaired electron. It is different from the OH hydroxide ion, which has two paired electrons and a negative charge to it. This molecule is stable. This one is not. And as I mentioned, all radicals, whenever they bump into another molecule, they'll steal electrons from that molecule and damage that molecule. Hydroxyl radicals is made by aerobic respiration in trace amounts. It's also made by ionizing radiation. And when hydrogen peroxide reacts with certain metal ions, our cells remove the hydroxyl radical with antioxidants. The antioxidant combines with the hydroxyl radical and then inactivates it. Some examples of antioxidants that act to remove hydroxyl radicals are actually vitamin C and vitamin E. Any questions about any of that? If there's no questions, we're going to stop here, and I will see you in the lab at 6:30. All right, goodbye.